Hi, my name's Todd Dunn. I have chronic lymphocytic leukemia. I'm also a scientist, specifically a retired professor of geochemistry and chemistry. As a chronic lymphocytic leukemia patient, I have a lot of blood tests. But as a scientist, I wanted to understand my blood tests a little bit better than the average lay person. So, what did I do? Well, I went out and bought a biological microscope and the necessary supplies to make blood smears so that I could look at samples of my blood under the microscope. And what I'm going to do today is show you the results of having a look at my blood. I'm also going to show you the microscope and explain a little bit about what the basic requirements are for a microscope to get good images of your blood. This is the microscope I bought. It's a pretty basic microscope, certainly not a high quality one by any means, but it's good enough to do blood work. There are a couple of basic requirements for doing blood work. First off, if you really want to be able to see the blood cells well, you need a microscope that has a 100 power oil immersion objective lens, and this one does. This microscope has 4 objective lenses, 4 power, 10 power, 40 power, and 100 power oil immersion. It also has a 10 power ocular lens, that's the eyepiece, so the microscope has 1000 magnification. The microscope also came with a digital camera, so I can connect it up to my computer, which is what I did to take the pictures you're going to see later on in this video. One important feature is visible right here. That's a mechanical stage. That's necessary if you're going to make smooth, regular movements of the slide on the scale that you need to be able to look at blood cells, which are quite small. Okay, now we're looking at underneath the stage. Right here is a substage condenser. That is necessary to get good, sharp images of small blood cells. So basically what we need is a microscope that has a fully adjustable substage condenser with an iris diaphragm, 4 objective lenses. Really you only need about 2, but one of them has to be a 100 magnification oil immersion lens. Ideally you'd like plan lenses, which will give you a full field of view and focus. This microscope, being cheap, does not have plan lenses. Someday I might upgrade it, we'll see. And if you want to transfer images to the computer, you really need a digital camera of some sort. I could have bought an adapter to put my digital single lens reflex camera on there, but that was more money and I didn't want to get too far into it. So that's the basic microscope that I use to take the pictures you're going to see of my blood. There are a couple of other things you're going to need if you're going to look at your blood under the microscope. First off, you're going to need some microscope slides. So I bought 100. You're also going to need some stains in order to make the blood cells show up better. On the left here is a stain called Eosin Y. That stain is what will make the red blood cells assume their characteristic pinkish red color under the microscope. And the other stain here is called methylene blue, and the methylene blue stain is used to dye the nuclei of the cells blue so that we can differentiate the different types of white blood cells. There's one other component that you're going to need, and that's methanol. Methanol is used to actually stick the cells to the glass slide, which is called fixation. I couldn't find high purity anhydrous methanol in laboratory grade locally, so I bought the next best thing. This stuff. This is heat fuel treatment. It's used for dewatering gasoline, and it says on the package contains methanol. So before I bought it, I looked up the material safety data sheet to find out what was actually in it, and it turns out it's 99.99% methanol, and it's dry methanol, which is exactly what I wanted. And it was cheap too. This bottle, which is enough to make dozens of slides, cost about $2.50. So these are the basic supplies you need. Now let's look at my blood. This is a photomicrograph taken at 100 magnification. The gray round cells you can see are red blood cells, and in this field of view there are approximately 1,100 of them. The dark blue cells that are nice and round are lymphocytes, and there are 11 of them. The lighter blue cells, which are more indistinct, are actually smudge cells, which are lymphocytes that were damaged during preparation of the blood smear. So, as I said, there are 11 lymphocytes, there are 3 smudge cells, and there are approximately 1,100 red blood cells in this slide. If I had a normal white cell count, there would only be about 1 to 2 white cells in this field of view. The fact that there are actually 14, if I count lymphocytes and smudge cells, suggests that I have a very high white count and specifically a high lymphocyte count. The presence of smudge cells is indicative of chronic lymphocytic leukemia. So this is a typical field of view for a person with a high lymphocyte count who has CLL. At this point, I'm going to move on and start talking a little bit about the specific cells that you might see when you look at a blood smear. The next few pictures are of individual cells, and they're all taken at 1000 magnification using the oil immersion lens. This first picture is a typical lymphocyte. In lymphocytes, the nucleus of the cell stains dark blue and the cytoplasm stains a pale blue. You'll notice in this cell that the nucleus makes up about 85 to 90% of the cell. That's pretty typical. In some cases you can't see any cytoplasm at all. You'll also notice that this lymphocyte is about the same size as, or maybe slightly bigger than, a red blood cell. Most of the lymphocytes, in my blood anyway, are about this size. There are a few that are a little bit bigger, but this is what a typical lymphocyte looks like. Very easy to identify. This is a typical neutrophil. You'll notice that this neutrophil is characterized by a multi-lobed nucleus. This one has 4 lobes at least, connected by narrow little bands of nuclear material and surrounded by cytoplasm. Notice this cell is at least twice as big as the red blood cells that surround it, which is pretty typical for neutrophils. They are bigger cells than red blood cells or my typical lymphocytes. These are very easy to pick out because of the multi-lobed nature of the nucleus. Here is another neutrophil adjacent to 3 lymphocytes. You'll notice that the neutrophil is much larger than the lymphocytes, and also you can really see the contrast in the nuclei of the various cells and see why it's very easy to identify neutrophils. This is a monocyte. It is a granulocyte, which means that it has granules in the cytoplasm, which you can easily see in this picture, where the cytoplasm is almost opaque from all the small granules. Monocytes also tend to have slightly less regular boundaries. You notice this is a little rough, not smooth like the red blood cells adjacent to it. And the nucleus is much smaller than the cell overall. So we're looking at granular cytoplasm, smaller nucleus, often with a small indentation in it. Those are the characteristic features of monocytes. You'll also notice it's quite a bit larger, at least twice the diameter of the red blood cells. But again, a relatively easy cell to distinguish from neutrophils and from lymphocytes. The other 2 types of white blood cells are basophils and eosinophils. They are much less common. They're the least abundant white blood cells. I don't have any pictures of them today simply because I didn't find any when I looked through this blood smear. The last thing I want to talk about today is platelets. The reason for that is that for people with CLL, platelet counts can be a trigger for treatment. This slide has 2 platelets in it. You can see the 2 lymphocytes. Above the upper lymphocyte there are 3 red blood cells, and the small cell just above those 3 is a platelet. Similarly, if you go to the lower lymphocyte, find a cluster of red blood cells below it, and then another platelet down underneath those cells. Platelets typically are present in your blood at about 7% of the number of red blood cells. So you would expect that for about every 15 red blood cells there would be a platelet. Clearly, in this slide, which is at 400 magnification, I have fewer platelets than that. That's because my platelet count is under 100,000. Another thing that you can do from a blood smear, in addition to simply identifying the types of cells that are present, is to determine what's called a differential count, which is basically to figure out the relative proportions of the different kinds of white blood cells. The way you do that is very simple. You drop your microscope down to 100 power, and you simply count all of the white blood cells in the field of view, noting what type they are. So in this field of view, there are 30 white blood cells. One of them is a neutrophil, and one of them is a monocyte. So there are 28 lymphocytes. You do that for a number of fields of view until you have accumulated the total number of white blood cells that you want to count to get the sort of precision you're thinking of. Typically, if the lab at your hospital does it, they're going to count 100 or 200 white blood cells. So the precision won't be very good. In my case, when I did this differential, I counted a total of 2,124 white blood cells. 2,089 of them were lymphocytes. I found 24 neutrophils and 9 monocytes. That works out to 98.4% lymphocytes, 1.1% neutrophils, and 0.4% monocytes. So that's a typical differential. If I knew my total white blood cell count, which you cannot determine from a blood smear, I would then be able to calculate my absolute lymphocyte, absolute neutrophil, absolute monocyte, etc. counts. To get accurate absolute counts, manually you have to use a special glass slide called a hemocytometer, which has known areas and a known depth that you count cells in, and then you can get an absolute count. Okay, I hope that this has illustrated some of the things you can see if you take a drop of your blood, put it on a microscope slide, stain it, and then have a look at it with a microscope. Anybody can do this if they have a suitable microscope. The only really hard part is getting the drop of blood, because you have to stab yourself with something so that you bleed. Okay, thanks a lot. I hope you enjoyed it, and I hope you learned something about your blood.